All right, welcome back from our break. Our next presentation is entitled A Sustainable Vaccine Ecosystem, Successes, Gaps, and Innovation Opportunities. The Economist Group's Vaccine Ecosystem Initiative strives to promote a sustainable vaccine system by examining and reimagining elements critical for vaccine development, deployment, and adoption. Building on this work, the initiative has created a new and innovative tool to understand country-level readiness to harness the full potential of vaccines, the Immunization Readiness Index. The index measures the enabling environment for equitable and sustainable immunization in 30 countries around the world. It assesses policies, strategies, and actions that facilitate both routine and emergency vaccination programs. In this session, we'll hear about the Immunization Readiness Index, including key findings, best practices, and opportunities for improvement. Our speakers are David Humphreys and Miranda Baxa from The Economist Impact. You have the floor.
All right, good morning, everyone. Thank you so much for the opportunity to participate in this wonderful session. Hearing your discussions over the last 2 days has been very informative for both Miranda and I as we take this work forward. The intent here is to share, as Bob mentioned, some of those latest results from the work we've done in vaccines, including the first ever Immunization Readiness Index that we've generated. And part of that goal is to be able to connect the dots with all the wonderful information that we've all collected over time related to immunization, related to health, and related to broader societal issues like climate change. So they're very much interconnected. And so the intent is to share some of those findings. I think it will very much resonate with the topics that you cover in innovation, in equity, in vaccine confidence. Before I do that, let's see if I can manage the slides here. Just a brief word on us and the ecosystem work that we've done. I would hope some of you know a publication that we run called The Economist. There's an interesting article this week about AI and science. I encourage people to read. Certainly is our flagship publication that we have, but The Economist Group has long participated in this space through the Economist Intelligence Unit, macroeconomic forecasting events like our cancer series that we've just run this week, and Economist Impact, which brings together our policy think tank, our creative storytelling, and our media channels to really engage, educate, and really drive change. And within that, we've covered a number of issues in food security and oceans health and financial inclusion and topics related to health, topics in cardiovascular, topics related to system and patient centricity, and topics around the connection of health and society, economic implications of health, the state of health inclusivity. It was great to hear the previous panel and how those all interrelate. And so part of our goal is to be able to educate and drive change and be part of dialogue here. And the concept of the ecosystem is a good example in the vaccines of our purpose and our work. And this started late 2020, right before the rollout of the COVID-19 vaccines. And we saw it as a window of opportunity, with all the attention being paid to vaccines for people and stakeholders looking at this. Could we leverage that moment to educate, to drive awareness, and build a more sustainable vaccine ecosystem related to COVID and beyond? And so that was the purpose of the program, to raise awareness around the issues of vaccines across lifehood, to be able to break down the issues that really matter and learn what are the levers that really drive change, to engage people like all yourselves around this in the dialogue and discourse, and from our perspective, see what may come next and what we may look and continue to generate our knowledge, and then raise and elevate the issue of the importance of vaccines in health and beyond. That connection to climate change and being prepared for what may become societal changes is immensely important. And so if we can make that connection, that certainly is one of the goals that we have. Now, the vaccine ecosystem here, and my colleague Miranda will walk you through the Immunization Readiness Index, which is a really exciting tool that we've gotten a lot of attention from a multitude of stakeholders around the world. This gives you a sense of the breakdown of the work that we did. Beyond the index that we looked at, we broke down elements of the ecosystem to try to understand the challenges, try to understand those lessons from COVID, and try to understand a future which engages those within health and those that are beyond, but participate in a more dynamic ecosystem. All the information that we'll talk about here is publicly available. You'll see a QR code for people to be able to see and find that, and hopefully people will be able to engage in that information. As part of this process, and I think from the previous subcommittee meeting that we had, our work was not just based on research analysis and engagement, many people like yourself, in an organization advisory council, and getting different experts from different walks of life in different parts of the world to say, how do we need to reimagine this? So many of the results, the framework, the analysis that's done is a reflection of their collective experience. We also think it's a great way to be able to have the multiplier effect and connect that information back into the world so it does have an impact. So I just want to briefly touch, before we get into the immunization ready index, a couple of interesting insights that came from our ecosystem work. As I mentioned, in the ecosystem, that consists of 5 interrelated pillars that goes from research development all the way to user acceptance and uptake. That was based on roundtable conversations we had, again, with people and entities within the world of health and outside, including those in technology firms, including supply chain, including community level organizations. So we include a wide variety of perspectives in trying to understand the way forward and breaking down those issues that really impact the ecosystem. Now, I want to give you a little bit of a snapshot of the results here. This is from the first pillar in R&D that we did. Not only does it generate analysis, but it has kind of consensus recommendations that came from our experts that you see on the left-hand side, directed to different stakeholders who are participating in this. And so in R&D, thinking from the earliest stage all the way to clinical trials and that process of regulatory systems, the technologies that really drive vaccines, and the infrastructure required to be able to deliver. And within that, some of the themes you already talked about today, that early stage investment in R&D, and embedding not only the issues and the threats we face, but embedding concepts like equity at the earliest of stages and have that nuanced view. It's also being able to adapt to the speed and the pace and the complexity of the vaccines to come, and making sure that we're able to maintain a safe and innovative environment, be able to take advantage of that potential. And part of that process is educating the public on those new technologies and what they need and new processes, so we get their buy-in along the way, and take advantage of the moment where people are very well aware of things like mRNA. And that leads into the last pillar there in user acceptance, and trying to understand those perceptions, the misinformation, information, where people get information, public trust, and a nuanced view, and engaging the mechanisms to drive that particular trust. And we heard some of the conversations and data, and trying to understand that using techniques from a marketing behavioral science of how do you engage different populations that behave differently, consume and trust information differently. The second piece, and it's a really critical element, not just within our vaccine work, but the work that we've done in pandemic response, the work we've done in equity, which is that multi-stakeholder collaboration, and particularly important at the local community level, because that is where people go to get trusted information. That's where they're able to go and to connect and communicate in a way that is effective. And so from a user piece, that's really very critical. With that said, I'm delighted to be able to pass over to Miranda here shortly, that she can kind of walk you through the top level findings. A couple words of caution when we talk about an index. An index is a tool that it really looks at the enabling environment, the policies, the strategies, the interventions that matter in a particular area, in this case around immunization. It is meant to be a cross-geography comparison, but it's not meant to look specifically at rankings. The details matter for us. The rankings may get headlines. The details within that comparison is important. It also could be a diagnostic tool. We've taken to many country stakeholders and engagements to identify what are the levers that matter for your particular circumstances, and how can you learn from others that have not only put up best practices and policies, but how they've actually sold those policies to other stakeholders within health and outside of health. It is also not meant to be necessarily a predictive tool. Looking at what we call inputs in the policies and outcomes like vaccination rates. So over time, we actually see a very strong relationship with a wide variety of areas. And one of the things we look to do in the future is start to understand those connections with broader societal things. Pass it over to Miranda so she can share you some of those results. Thank you again for your time.
Thank you. As David mentioned, the Vaccine Ecosystem Initiative explores the components of the global vaccine ecosystem. But as an extension of this work, we've also created a new tool for understanding country-level readiness to harness the full potential of vaccines. and that is the Immunization Readiness Index. So the index measures the enabling environment for both equitable and sustainable immunization in 30 countries, including the US. And it assesses the policies, strategies, and actions that are necessary for facilitating both routine vaccination programs and those for public health emergencies like COVID-19. And this tool allows us to make comparable country-level assessments of the policy landscape that underlies immunization efforts so that we can identify opportunities where countries might make improvements. The index includes 50 indicators across 6 domains, and these evaluate the policies and processes that are involved in creating robust and resilient immunization programs. Each of the domains covers a distinct action area that is necessary for supporting the vaccine uptake pathway with the ultimate goal of getting shots in arms. These domains are inherently interconnected, and they are also aligned with the pillars of the global vaccine ecosystem. But in contrast to the pillars that David has described, these are components that countries need in-house in order to sufficiently support and to achieve immunization readiness. So the first 4 domains reflect a chronological process that starts with developing a national immunization strategy and approving new vaccines, through to ensuring that vaccination programs are sufficiently funded and that there is an adequate delivery infrastructure for actually bringing the vaccines to the population. Then domain 5 includes concepts related to building public trust in vaccines and reaching all members of the population through outreach activities. And when done successfully, these activities will take place throughout the sequential stages represented by the first 4 domains. And finally, domain 6 provides context to the others. So these are societal and governmental factors that are not necessarily indicative of a country's level of readiness on their own, but they are important for understanding whether the national environment is conducive to immunization efforts. As David mentioned, our primary goal in creating this tool was really to identify substantive actions in areas that could lead to country-level improvement. And so we, as a research team, decided not to formally rank countries, but instead countries are grouped according to their overall performance into 6 tiers representing varying degrees of readiness, with tier 1 representing countries that are the most ready and tier 6 countries that are the least ready. And countries tend to perform well in the first 2 domains related to policy planning and regulatory mechanisms. So we know that they have the necessary foundations for immunization activities, but what they are able to write in policy is not always successfully translated into practice. And so we see much lower performance in the latter domains related to procurement, delivery, and outreach. And this disconnect between policy and practice exists in most of the countries that are included in the index. So we know that there is really more work to be done in order to effectively execute what is set out in national immunization strategies. Ensuring equitable access is another critical part of achieving immunization readiness. So we've highlighted a few indicators related to equity to help countries benchmark their progress towards achieving equitable immunization for all. You will notice that countries that have better rates of immunization readiness, like the top 10 performing countries, tend to demonstrate a greater focus on equity when creating and carrying out their national immunization strategies. And the US does very well in this regard, satisfying the requirements for all 4 of the equity indicators. But even for countries with lower levels of immunization readiness, like our bottom 10 performing countries, we still see evidence of equity-focused action, illustrating that countries are recognizing the importance of health equity in immunization. Now that I've given you a bit of background on the index overall, we can dig into our evaluation of the US. So the US, of course, has an established immunization program and a history of meeting immunization goals. So it's no surprise that they perform well and above the overall average level of performance. for overall immunization readiness and across domains. The U.S. performs best in Domain 2, regulation, and that includes indicators related to approving new vaccines, participating in regional and international data sharing, and engaging with R&D and manufacturers. And as a matter of fact, FDA's engagement with industry during COVID-19 as part of Operation Warp Speed is featured in our report as a best practice example of multi-sectoral cooperation. Even so, there are some opportunities for improvement, and those are particularly in Domain 5 related to outreach, which is the only domain in which the US performs below the average level for high-income nations. So we've also highlighted a few specific opportunities for improvement. And the first is in Domain 5 that I just mentioned, and that is the existence of a national risk communication strategy for vaccine preventable diseases. While HHS, CDC, and others do provide relevant guidelines and information, there's no evidence of a formal national strategy for this. And there are countries in varying stages of development in the index that do have clear risk communication mechanisms for diseases that can be prevented by vaccines. And we know that these strategies can be effective in helping populations understand why vaccines are important and therefore improving uptake. The US has had successful risk communication campaigns in the past. For instance, with the We Can Do This campaign during COVID, but we would love to see this codified in policy so it can be leveraged in the future for emergency and routine vaccines and diseases. Another area for improvement is in Domain 4, delivery, and that is related to data on health worker competency in immunization. With this indicator, we are not assessing whether or not health personnel are trained or have the competency, because we know that they do. But rather, we're concerned with whether or not this information is being collected and made available in the public domain. And very few countries actually do this. In fact, none of the high-income nations that we've included in the index have met the criteria for this indicator. But we think it is very important, not only for transparency and accountability, but also for ensuring that countries have sufficient healthcare infrastructure and healthcare workforce for immunizing the population. And it can also be helpful in making sure that we are prepared for future public health emergencies. Recommendations and best practice examples for each of the domains are included in our report, but I'll leave you with some key takeaways that emerged in our analysis of the US. So first, successfully translating policy into practice is key to achieving immunization goals. While the policy is important and a necessary foundation for immunization programs, it's really the operationalization of that policy that gets shots in arms. And for the US, the delivery and outreach domains present the most opportunities for improvement. Increasing support for state immunization programs through additional funding and more detailed guidelines can help states to better prepare to increase vaccination coverage and make sure that they are reaching all of their residents. And finally, codifying the concept of equity in policy and prioritizing equitable practice will help to ensure that all people benefit from immunization. As I mentioned, the US already does this really well, but we encourage you to keep the momentum going and to keep equity top of mind when creating new policies or programs that seek to improve immunization. Diseases do not recognize state or national borders, nor do they stop at the doorway when we go into our homes. And so to make sure that we are adequately protecting the health of our families, our communities, and our nations, we have a responsibility to make sure that everyone is protected. Thank you again for the opportunity to speak with you today. We are really honored to be here and hopeful that this information will be helpful to you as you try to make improvements in immunization. So we encourage you to take a look at our website. There is a QR code there, but it appears as though it's been cut off. So the website is vaccineecosystem.economist.com, and that hosts all of the data I've shared with you today and more, and has our report and data dashboard available for download. We are happy to take any questions that you might have, either about the Vaccine Ecosystem Initiative or the Immunization Readiness Index. Thank you again.
Thank you both. Miranda, you said that was vaccineecosystem.economist.com. Is that correct? Okay, good. Just wanted to clarify that. Any questions or comments from the committee members here in the room or online? I certainly, in discussions with a number of our members over the last several years that I've been a part of this group, think that many would echo the need to continue to support our state and county and local health authorities and units in trying to get those outreach efforts out, to get things, particularly in some of our deserts of healthcare, in our rural areas and in some of our urban areas also. Rebecca Quo.
Thank you for such a really interesting presentation. I guess my question is, looking at the US, we have a lot of states and jurisdictions that operate somewhat independently. I'm curious, in your evaluation or assessment, did you look at the state level, or was it strictly at the national level? And then I guess there's a second part. If you looked at it, or if you didn't look at it at the state level, is there ability to take parts of your assessment and apply that to the state level? Thank you.
We primarily looked at national policies. This is a global index where we are considering other countries, and so we took a national approach. We also know that it is important to have these policies and programs situated nationally as much as possible, so that we can make sure that things are happening equitably within the country and that there aren't extreme differences between states or jurisdictions, which is why part of the reason that we take that approach. There hopefully will be an opportunity to add some subnational or state information going forward. David, do you want to speak to that?
Yeah, we've had a lot of interest, particularly in certain states here, to apply this as a diagnostic tool, essentially identify what do we do next. And in many cases, major countries around the world, US, Brazil, China, India, where there's just so much variety and autonomy, doing that sub-level national analysis actually makes a lot of sense. And so our hope is one of the extensions of this piece of work is not just to expand number of countries, but actually dive deeper. All right, and you can look at it through many different lenses. The geographic lens is important. What about the community lens? What about the difference between cities and rural environments? That could be really quite interesting. The whole point is to drill down into the levers that matter and to get people to focus, whatever their efforts may be, on the things that are going to drive change.
Rob Schechter.
I'd like to echo the appreciation for your presentation and all the work in the models and the index. A couple of questions. One is, were there characteristics, common denominators between countries with strong outreach scores or outreach programs that would be helpful for us in our efforts? And the second question is, are coverage rates reflected in or correlated, are they either reflected in or correlated with the index?
Thank you for that question. So to your first question related to countries that have already done well in outreach, one of the things we noticed was that these countries tend to actually go out into hard to reach areas to engage with people that either, or communities that either have high rates of zero dose children, to engage with them, provide education, and actually vaccinate those populations. And they also seek to understand reasons why particular areas might have lower rates of coverage. So for instance, sitting down with vaccine hesitant individuals to try and understand their specific concerns and what they need in terms of support or information in order to encourage them to be vaccinated, similar to what some of the earlier presentations have shared with you as well. Your second question, can you remind me?
Yes, whether immunization coverage rates are either reflected in the index or correlated with the index.
Thank you. They are correlated with the index somewhat. There's a moderate positive relationship. But one thing that I will say about the vaccination rates, there's been a lot of variation over the last 40 years or so. This is not specific to the US, but we see this globally. Immunization programs are really susceptible to outside pressures. And so we're seeing that now in the wake of the COVID-19 emergency as well, where there are much higher rates of zero-dose children, and there's been a lot of fluctuation in vaccination rates. And so now is not a great time to use vaccination rates as a way to correlate, nor would we want current vaccination rates to really be reflective of what we're seeing in the policies and programs. And so we're hoping as we build on this index, collect more information, and also as countries are able to rebound from the pressures of COVID-19, we will see a higher correlation going forward.
John Iskander.
John Iskander, Defense Health Agency. So the focus on healthcare worker competency and immunization caught my attention. Do you all have recommended best practices? Is the focus on knowledge, on competency, on demonstration of abilities, and again, similar to what others have asked, are there best practice countries we can look to?
There are a few countries that make the data available that I mentioned earlier. Surprisingly, or maybe not surprisingly, these are countries that have newer immunization programs and strategies that have ramped up significantly over the last few decades. So countries like Kenya, for example, have really good data on this. But high-income nations, like I said, really just don't make this data available at this time. And we would like to see that available primarily for transparency and accountability, but also for preparation. But in terms of the actual competency and training, one of the things that we have found and think is really important is a cultural competency training. So if you are sending healthcare workers into rural areas, for example, understanding what the needs are of those specific communities. If you are sending healthcare workers to vaccinate people with disabilities, what are their needs? What are their concerns? Or vaccine hesitant individuals? And so we need to take less of a one-size-fits-all approach when it comes to training our healthcare workers to go out into the field and vaccinate populations. Thank you.
Well, thank you very much for your presentation. Just one thing to think about. You mentioned to Rob Schechter's question earlier about correlation with vaccination rates. You might think about whether there are correlations with Delta or change in vaccination rates for children and for adults as a reflection on the stability of those elements that go into the to your index. Thank you.
Yeah, it is something we've considered, quite honestly, and I think we'll include not just a composite index around child immunization. We also look at issues of equity, right? The national level is important. What does the disparity exist across different jurisdictions?
Well, thank you all for your presentation.
Thank you.
We're now going to move to our next panel. Our next panel is entitled Unfinished Triumphs, Vaccines and Development, and I'll invite the speakers that are in the room to come to the stage. Traditionally, vaccines prevent disease, but therapeutic vaccines work by stimulating the immune system to treat a disease or slow its progression. Currently, only 3 therapeutic vaccines, all of which treat cancer, have been approved for use by the FDA. However, there are exciting therapeutic vaccines in development to treat diseases such as hepatitis B, herpes simplex virus types 1 and 2, human papillomavirus, syphilis, as well as cancer and other diseases. In this session, the presenters will cover several therapeutic and preventive vaccines in development. Our presenters for this panel are Dr. Hildegard Ertl from the Wistar Institute, Dr. Vinod Balachandran from Memorial Sloan Kettering Cancer Center, Dr. Harvey Friedman from the University of Pennsylvania, and Dr. Lorenzo Giaccani from the University of Washington. And I'll invite Dr. Ertl to take the podium. Your slides are up and you have the stage.
Okay, I hope I get this right. So this talk, thank you for having invited me, is about a therapeutic vaccine against chronic hepatitis B virus infection. And the results I will show you are based on mouse data. We are starting a clinical trial next month, so next year I can hopefully tell you about human data. Okay, we have a preventative vaccine against HPV, which is extremely efficacious. Nevertheless, it's underutilized, so chronic HPV remains a huge global problem. More than 250,000 people, 290 million people have chronic HPV, and most of them are undiagnosed, and nearly all of them are untreated. There are treatments available in form of drugs. They will suppress HPV loads, but they will not cause a cure. So they have been taken for the lifetime of an individual, which for many countries is simply not a financial option. So the vaccines we came up with are based on chimpanzee adenoviral vectors, and since COVID-19 and AstraZeneca, which use related vaccines, we know all about them. We are using inserts based on HPV polymerase and core, and I'll explain that in more detail, and we have a genetic adjuvant. So basically the inserts we compared the sequences of core and polymerase of genotypes A and B from over 8,600 published sequences and selected regions of conservation. And then we checked these regions for epitopes from multiple HDA genotypes and selected segments that were rich in these epitopes. And then we made initially 3 vaccines, one that has the C terminus of polymerase, one at the N terminus of polymerase, and one at core. Based on these conserved epitope-rich sequences, we test them in mice quite extensively, and then we made a single gene where we combined the most immunogenic regions of these 3 inserts. The genetic adjuvant, which we think is key, is the herpes simplex virus glycoprotein D. So when T cells become activated, they recognize with the T cell receptor a peptide expressed by an antigen-presenting cell. Is there a pointer on here? I guess not. So at the same time, when there's activation to the T cell receptor and co-stimulators such as CD28, there's already inhibition to prevent an overwhelming T cell response. And this inhibitory pathway is called the B and T lymphocyte attenuator, BTLA. And that binds to the herpes virus entry mediator on antigen-presenting cells, and that basically dampens T cell receptor signaling. HVEM also binds a coactivator, LIGHT, which in a trimolar complex of LIGHT, HVEM, and BTLA is suppressed. Then inhibition prevails. What GD does, it has structural similarity to BTLA as shown in the middle. It can bind to the BTLA binding site on HVM, but it binds with higher affinity. So it blocks BTLA binding and thereby prevents the dampening of the T cell receptor signaling. So you lower the threshold for T cell activation and you get a higher response and a much broader response, including response to subdominant epitopes. So what we are doing, we are putting this insert, which is composed of GD and our HPV sequence away from the HVM binding site into an adenoviral vector. The adenovirus transduces antigen-presenting cells. The protein is produced, some of it is misfolded. It's chopped up into peptides presented by the T cell by MHC to the T cell receptor. Some of it is presented in full length. It has a signal sequence, thanks to GD. It's going to the cell surface. It stays there, for it also has transmembrane domain. And then it blocks the BTLA HVM binding and amplifies the T cell response. Okay, wrong way. So this is basically the T cell response we get in blood after we immunize mice. This is percent interferon gamma producing cells, or CD8 cells over all CD8 T cells tested at 2 different vector concentrations after 4 or 8 weeks. And you can see we get a very robust CD8 response, which goes up between weeks 4 and 8. There's not a really strict dose response effect, and we see about a frequency of 4%, which is a good frequency. As we selected for CD8 epitope-rich regions, they are not epitopes, they are segments, we don't really get a particularly good CD4 response. Then we looked at the breadth of the T-cell response. We basically used splenocytes from mice that had been immunized and tested them against each peptide that's in the sequence of the insert. Each peptide is 15 amino acids in length and overlaps by 10 amino acids with the adjacent peptide. On the upper part, you see the GD-HBV2 vaccine, and you can see that a lot of epitopes are being recognized. The same vaccine without GD, there's also quite a number of epitopes that are being recognized, so we selected for epitope-rich regions. But if you look at the number of epitopes, the breadth is by far higher if we include GD. It goes up from 34 peptides to 51 peptides. Then we look for efficacy. HPV does not infect mice. Chimpanzees, which are natural hosts of HPV, are out of the question to be used. So we used a surrogate model, which is far from perfect, but the best we can do. We used an AAV8 vector, which expressed the over-length HPV genome. What AAV8, which stands for adeno-associated virus, does, it's very hepatotropic. So if you inject into the tailbone of a mouse, it goes to the liver, and that mouse will now make HPV virus particles. So when we inject these mice with AAV, they start making AAV genome copies, which we can test for in blood. These mice were vaccinated 4 weeks later with the vaccine. They express either the GD-HBV2 or the HBV2 without GD. And then periodically we will bled the mice and test them either for genome copy numbers by qPCR or for T-cell responses by intercellular cytokine staining. And as you can see, if we don't vaccinate the mice, the titers that were around 10 to the 8 at baseline go up slightly to 10 to the 9 and then stay more or less stable. If we immunize with GD-HBV2, we see a nice 2 to 3 log decline in titers. If we use HBV2 without GD, we see about a 1 log decline, nothing impressive. And as you can see, we get a very robust T-cell response in liver. We wanted to make this experiment more stringent, so we went up the AAV dose to 10 to the 10. Otherwise, the experiment was identical. And as you can see, with this higher vector dose from baseline, which is 4 weeks after AAV injection, till the end of the experiment, there is a substantial increase in HPV genome copy numbers in blood. With the GD-HBV2, there is no increase, there is 3 log decrease. With HBV2, there's again a slight decrease. Not only do we decrease the genome copy numbers of HPV, we also decrease surface antigen levels. Also, our vaccine does not express sequence of the S antigen, that's shown over there. Actually, 2 mice, we got levels down to below detection. And again, we see a very robust CD8 response, not just in the spleen, which is shown in the lower left panel in white, but also in the liver, which is shown in gray. And if we look at correlations between genome copy numbers and CD8 T-cell responses, there's a very good inverse correlation. The higher our CD8 responses are, the lower our genome copy numbers are. And that's both the case for liver and spleen T-cells. So in conclusion, this is an ongoing experiment. We want to increase the stringency even further. So we vaccinated 4 weeks after infection. That's not really what's happened in humans. They are infected for years. A mouse doesn't live for years, so we waited 4 months, which is about a sixth of the lifespan of a mouse, equivalent about 12 to 14 years in humans. This is the right slide. So this is an ongoing experiment, so we are not at the end point yet. As you can see, we waited till the HBV genome copy numbers had stabilized around week 16. Then we vaccinate the mice, and we used a control vaccine, which expresses some melanoma antigens in blue. There's absolutely no change in genome titers. We used the GD HPV2 vaccine, and you can see this time with a slight delay, there's again about a 2-log drop in titers. And again, our T-cell responses are quite high. So in conclusion, the GD-HPV2 vaccine elicits a potent and very broad CD8 T-cell response against HPV antigens, which is polymerase and core. The T-cells not only can be detected in blood and spleen, but we also find them in the liver. When we use a surrogate challenge model, we see a very substantial HPV genome decline, which inversely correlates with CD8 T-cell responses. We also see a decline in HBs antigen. And again, there is a correlation with the T-cell response. And we are planning on a phase 1b trial, which will be initiated Q1 to 2 was aspirational, sorry, the slide is outdated. It's actually going to start next month. And we are using HPV-infected individuals that are virally suppressed due to drugs. And we're using a prime only as well as a prime boost. And we obviously will follow the usual safety standards. We will follow liver enzymes. We will follow T-cell responses. We will follow HB genome copies, HBs antigens, and all of this. And this work was mainly conducted by these 2 investigators in my lab, and funding was provided by Viral Therapeutics. Thank you.
Thank you very much. Our next presenter is Dr. Vinod Balachandran from Memorial Sloan Kettering Cancer Center. Your slides are up. You have the floor, sir.
Thank you very much for the opportunity. So a few months ago, many of you may have received this breaking news that a team at Memorial Sloan Kettering Cancer Center, this was our team, had completed potentially an important breakthrough in one of the most challenging cancers in all of oncology, pancreatic cancer, by completing the first clinical trial of mRNA vaccines for pancreatic cancer, and had discovered through this clinical trial that mRNA vaccines could stimulate strong immune responses in half of these pancreatic cancer patients treated who generated an immune response. These patients showed no signs of recurrence of their cancer during the course of this clinical trial. So today I'd like to share a little bit about how we got here and some forward-looking thinking on how we can use this potential breakthrough to now expand cancer vaccination to other high unmet need cancers. So pancreatic cancer is a deadly cancer, soon to become the second highest leading cause of cancer death in the United States by 2025. So in only another 2 years, more cancer deaths from pancreatic cancer from many other common cancers, such as colon cancer, breast cancer, prostate cancer, ovarian cancer, second only to lung cancer. And this is because the current treatments for pancreatic cancer, which include surgery, chemotherapy, and radiation, are largely ineffective, leaving current survival rates, despite these best current treatments, at only approximately 12%. And this survival rate, despite our best current treatments, has remained largely unchanged for nearly 50 years. So really highlighting the urgent need for novel treatments for this deadly cancer. But fascinatingly, although the majority, about 88% of pancreatic cancer patients who receive standard treatments live less than 5 years, a rare 12% of pancreatic cancer patients survive long term. And they receive the same treatments as all else. Why? Unknown. So about a few years ago, we started a program at Memorial Sloan Kettering to study these rare long-term survivors of pancreatic cancer with really 2 questions in mind. How do they do this, and how can we replicate this therapeutically? And when we started this program, this was back in 2015. And 2017, what we discovered is one of the most striking observations in these rare long-term survivors of pancreatic cancer was their tumors had approximately 12 times greater densities of activated CD8 T cells in their tumors. And what was also quite striking about this observation is not only were their tumors enriched in activated T cells, we could identify the exact same T cells that were found in these patients' tumors persisting and circulating spontaneously in their bloodstream up to a decade later, suggesting perhaps this was an auto-vaccination. So this led us to the question of if this is the case, then what are the antigens? Because perhaps by identifying the antigens, we can then deliver these antigens to other pancreatic cancer patients in an effort to essentially phenocopy this long-term survivor state. So this led to a series of additional studies where we discovered that these antigens were in fact generated in mutations that were interestingly individual to every single patient's tumor arising in passenger mutations. So now raising an important therapeutic challenges, namely what would be the optimal strategy to select these mutations which are putative targets of the immune system. As you know, cancers can have many mutations, so which of these mutations are in fact the immunogenic ones and thus suited for a vaccine? And secondly, as these mutations were individualized for or individual to every single patient's tumor, this required a vaccine platform that will allow for rapid custom cancer vaccination in real time. So how do you make a custom cancer vaccine fast? So to answer these questions, we first developed a model to be able to select of the entire pool of mutations in a patient's tumor, which are the fraction of mutations that we think are in fact immunogenic and thus suited for vaccines. And to answer this question of how to custom vaccinate, we reasoned the most straightforward technology to allow for encapsulation of multiple antigens that are now included in a patient's tumor DNA is to now encapsulate this into strands of mRNA. And in 2017, we selected mRNA vaccines as our optimal vaccine delivery platform, and then we forged a collaboration between our group and the then not so well-known biotechnology company BioNTech to make mRNA vaccines for pancreatic cancer, a lowly mutated cancer historically thought to not contain such mutation-derived antigens suitable for vaccines. And we launched this first clinical trial to test the individualized mRNA neoantigen vaccines for pancreatic cancer, a lowly mutated cancer historically thought to not contain mutation-derived neoantigens suited for vaccines. And this was an investigator-initiated, a single-center phase 1 clinical trial where we targeted to accrue 20 surgically resectable pancreatic cancer patients on whom we performed surgery to remove their primary pancreatic cancers in New York. And then within 72 hours, we shipped their tumors to colleagues in Mainz, Germany, who then sequence the tumors, identify the mutations that are suitable for vaccines, and then custom manufacture individualized RNA vaccines for every single patient. These vaccines are made that including up to 20 class 1 and class 2 restricted neoantigens. These are delivered as 2 uridine mRNA decatopes in lipoplex nanoparticles intravenously. Now, while this is ongoing, back in New York, we benchmark patients to receive a single dose of PD-L1 blocking antibody or atezolizumab, and at 9 weeks after surgery, the first of 8 consecutive priming doses of their custom vaccine, followed by standard of care modified FOLFIRINOX chemotherapy, followed by a vaccine booster. Now, we started our clinical trial and treated our first patient in December of 2019, and shortly thereafter found ourselves conducting this complex clinical trial in the midst of the pandemic, which posed several unique challenges, not to mention clinical and research shutdowns, particularly in New York, which was the epicenter of the trial during March when this clinical trial was open and running. We also had to deal with difficulties in supply chain disruptions because this involved a cross-Atlantic transfer of patient material and drug in real time. And of course, now BioNTech had the added small distraction to make mRNA vaccines to save the world from COVID. But thankfully, despite this, given the infrastructure and teams that we have in place, we were able to not slow the trial down, but in fact accelerate the trial to complete it, despite the pandemic and these challenges, a full year ahead of schedule. And also, during the process, accumulate further evidence that the immune system plays an important role in these long-term survival or the exceptional outcomes of these pancreatic cancer patients. Okay, so in this trial, though we targeted to accrue 20 surgically resectable pancreatic cancer patients, we ended up needing to treat only 19 patients with surgery followed by atezolizumab, of which 16 received vaccines. So overall, only 1 of 19, or 5%, had vaccine non-manufacture. Okay, is rapid custom cancer vaccination feasible? Short answer is yes. You can see here on your left, our achieved time to atezolizumab was within 1 day, and more importantly, achieved time to vaccination within 3 days of benchmark times. So rapid custom cancer vaccination is feasible, fully integrated into a standard clinical workflow, and even after complex oncologic surgery, as is required for pancreatic cancer, and despite having to do this in the midst of a pandemic. So if we can do it like this, we can certainly scale this to other cancer types. Okay, do individualized mRNA neoantigen vaccines stimulate T cells in pancreatic cancer, a lowly mutated cancer historically thought to not contain mutation-derived neoantigens suited for vaccines? So to answer this question, we use 2 tests to monitor for vaccine-induced neoantigen-specific T cells. A primary test, which is standard ex vivo interferon gamma ELISpot, and a confirmatory test that we developed in our lab that identifies vaccine-expanded T cell clones using a TCRV beta sequencing, a assay that we call Clone Track, both assays that preferentially detect high magnitude vaccine-induced T cell responses. And what we find with these 2 criteria is that we designate patients to be a vaccine responder if they have a positive primary test and a positive confirmatory test as adjudicated by teams that are blinded to both the clinical results as well as the assay results. So you have to be a responder, you have to have a positive primary and a positive confirmatory. And with these criteria, we find that individualized mRNA neoantigen vaccines stimulate high magnitude T cell responses in 50% of unselected pancreatic cancer patients. We find that these vaccine-induced T cell responses are quite substantial in magnitude, reaching as high as 10% of all peripheral blood T cells, and appear to be durable, lasting as high as 2.5% of all blood T cells, even 2 years later, despite a post-vaccination chemotherapy. When we look at a median follow-up of 18 months that extended beyond our pre-specified secondary endpoint of 18-month recurrence-free survival, we find that vaccine responders have a median recurrence-free survival that's not reached compared to vaccine non-responders who have a median recurrence-free survival of 13.4 months, suggesting that vaccines may delay recurrence of pancreatic cancer. And as to follow up on this, we have a randomized phase 2 clinical trial that is now global and now open and enrolling at Sloan Kettering. But really more broadly, what is really the significance of this? Well, we think that the significance of this is this discovery, along with other recent improvements in vaccine technology, can help us unlock the how for cancer vaccines, which the community has always felt is possible, but perhaps we just didn't have the exact right way to be able to do this. And by doing this in one of the most challenging cancers in all of oncology, pancreatic cancer, perhaps this can provide important learnings that would now allow us to more effectively generate vaccines to prevent and treat other common deadly cancers. How do we do this? We think this requires active iteration between a discovery and therapy, fully in human, on three critical topics, which is really essential for an effective vaccine, namely selecting the optimal antigen, selecting the optimal tumors in patients that would be suitable for such a vaccine, and the optimal platforms that can generate high magnitude immune responses. And for this committee, what would be the current barriers that we really face for this right now? Well, some of the barriers are right now, we do not have a cost-effective centralized source of clinical-grade vaccines for academics. So for us to be able to do this at speed and scale, the technology is really not available for anyone in academia. So if you want to run a phase 1 clinical trial based upon your research, we do not currently have a way to be able to do this. There's also no open access to clinical-grade vaccine manufacturing technology. This technology, if made openly available throughout the country, I think this would really help spur efforts across the country and the world on how we can really harness this exceptional RNA vaccine technology for the purposes of cancer treatment. We also have insufficient mechanisms to spark further collaborations between academia and industry. I think this would be another way for us to be able to spur the field on. And finally, insufficient funding in vaccine discovery and application research. I think one of the major breakthroughs of the last 2 decades is that the optimal targets, antigenic targets suitable for cancer vaccines has now been discovered, to those arising in mutations. So now if we can find the antigens, and we have potentially an application platform that can allow for rapid custom cancer vaccination and generate strong immune responses, this really allows us to make rapid progress in our broad vision and goal to take vaccines that can both prevent as well as treat cancer. So with that, I'd like to highlight everyone in my lab who helped us with the work, and thank you all for your time and attention.
Thank you very much. We'll now move to the next member of our panel, Dr. Harvey Friedman from the University of Pennsylvania. Dr. Friedman, your slides are up. Friedman, I don't see you on the Zoom. Now I see you. Dr. Friedman,
Okay,
your
so
slides are up.
I'll start over again.
Okay, we hear you loud and clear. You have the floor, sir.
All right. My topic is our vaccine development for genital herpes. I think I'm going to need your help advancing slides. So I'll say next slide. See if that works.
That'll work great. Thank you, sir.
All right. Next slide. Outline. I'll discuss the rationale for our genital herpes vaccine. Mention some efforts previously that have not succeeded. Talk about the design of our candidate, which is a prophylactic vaccine, and then discuss what we're doing to come up with a therapeutic treatment vaccine as well. Next slide. 500 million people have genital herpes caused by HSV2, 500 million. Another 150 million have genital herpes caused by HSV1, placing genital herpes as the second most prevalent sexually transmitted infection. And what's the problem with genital herpes? Well, many, but the biggest one that I see and hear are concerns about transmission to intimate partners, and concern from a birthing mother, transmission to her newborn infant, shown in the bottom right in that picture. Now, probably from a public health point of view, the most important concern is that about genital herpes is that it predisposes to acquisition of HIV by about a factor of 3, and for transmission for those already infected. We're thinking the target population for a prevention vaccine eventually will be similar to HPV, adolescents before onset of sexual activity. But the initial studies will be done in young adults or adults in general. Okay, next slide. There's been 3 publications about major prior trials, all done by Big Pharma. They all used HSV glycoproteins, and the picture on the right shows the numerous glycoproteins that are on the envelope of the virus. They all use glycoprotein D, which initiates virus entry. One of them also used glycoprotein B, which is the fusion molecule for virus entry. GD starts it, GB, baby, finishes it. These vaccines were successful to some extent, but they did not meet the primary endpoint. And by some extent, I mean in one of the studies, it delayed onset of genital herpes compared to the control group by 5 months. In another study, it protected a subset of subjects, women that were double seronegative to HSV, they didn't have HSV1 or HSV2. And in a third, it protected against genital herpes caused by HSV1, but not HSV2. So based on these trials, I think it's fair to say that targeting entry molecules alone may not be sufficient. Next slide. Our approach has 2 novel components to it. The first is that we're trying to interrupt some of the immune evasion strategies of the virus. And I point out here it's glycoprotein C and E. Now, we can do this because these glycoproteins are expressed on the virion envelope and potentially amenable to antibodies that can bind and block their function, their immune evasion function. They also get expressed at the cell surface. Now, the cartoon I'm showing here, I don't know if you can see my pointer, but I'll say it, the glycoprotein C, GC, binds complement component C3b. So this is its immune evasion potential. C3b is the central component of the complement cascade, and by binding to this complement protein, the glycoprotein protects the virus or virus-infected cells from complement attack. In the right-hand part of the slide, I show an antibody molecule binding to an antigen. Here it's shown as glycoprotein D, but it could be a different antigen. The key point is that the right side, the Fc end of the antibody binds to glycoprotein E, and that acts to block the activities mediated by the Fc end of the antibody. These activities include antibody-dependent cellular cytotoxicity and complement activation. So GE, glycoprotein E, binds the Fc end of an antibody, including an antibody that's binding by its FAB domain to an antigen, and blocks the important activities mediated by this and by the antibody. So this cartoon represents probably about 20 years of work in my lab to show the evidence to prove these points. Now, what about vaccine? Well, the goal with the vaccine is shown on the right. We want to induce antibodies to the glycoproteins, to glycoprotein C, to bind to glycoprotein and not allow it to interact with complement C3b. Bind to glycoprotein E on the right side of the slide. Not allow glycoprotein E to interfere with the activities of the FCN. And since GD, glycoprotein D, is part of our vaccine, we're borrowing from previous studies, bind to that glycoprotein and block virus entry. Okay, next slide. Now, the second component that is novel, and it's not as novel as it was when we started in 2016, is to use nucleoside-modified mRNA. Now, nucleoside-modified mRNA, shown in this picture, it's the 3 glycoproteins in a lipid nanoparticle. The modification is methyl pseudouridine. That compound replaces uridine in the mRNA. And what does that do? Well, it avoids triggering innate sensors and TNF-alpha that are pro-inflammatory. It enhances the translation of the mRNA, so more protein is made. It's a strong stimulant for T follicular helper cells that help our B cells make high antibody levels and produce many high levels of memory B cells. So modified mRNA is an important component of this discovery that my colleague Drew Weissman and Katie Carrico had at Penn that led to the COVID vaccines. It's also incorporated in a lipid nanoparticle. And the lipid nanoparticle has two main functions. It delivers the mRNA, but it also serves as an adjuvant to enhance immune responses. Okay, next slide. Now, we use two models, mice and guinea pigs. Mice are for our vaccine studies. Mice get acute genital disease. They establish latency, which is the hallmark of herpes infections, but they don't develop spontaneous recurrences. So that's a shortfall. But they are great for studying B and T cell responses because the reagents are available. On the other hand, guinea pigs not only get acute disease and latency, but they have recurrences. They get recurrent genital lesions, and they shed HSV2 DNA in their genital secretions, much like humans. But we have very few T cell reagents, but we combine these two tools to study vaccines. Next slide. Next slide, please. Okay, I'm going to show one experiment where we looked at the ability of our trivalent vaccine, mRNA, to protect when challenged in guinea pigs 1 month after the second dose, or next slide, 8 months after the second dose, which is about 25% of the lifespan of the animal. So in the 1 month experiment, we immunize twice at 0 and 1 month. We bleed the animals for antibody studies before challenge. We challenge with a 25 LD50, which is a very potent challenge. And we follow the animals for genital disease and HSV2 DNA shedding as markers of a breakthrough infection. In the 8 month experiment, we're looking at the same things, and we compared it with mRNA with protein. And I can't see it on my slide. I'm not sure if you can, but the protein was the same parameters, except we used 3 immunizations, which is our routine at 2-week intervals instead of 2 immunizations at 1 month intervals. Okay, next slide. Let's look at how they did. So let's first at the 1 month challenge. So the first column that I've highlighted in the red block is the mRNA vaccine group. And then beside it is the protein group. If you look at neutralizing antibody titers, very high titers with the mRNA, 6,000. Good titers, very good with protein, but not quite as good. No deaths in either group. Genital disease, none. No genital disease developed in this challenge. Did they shed any DNA in their vaginal secretions? 1% of the days the guinea pigs had some shedding. But none of the guinea pigs actually shed infectious virus. It was DNA, but no virus. The protein did well, but not quite as well. You can see that on the right-hand side. Now let's look at the next slide, 8 months. Now at 8 months, the mRNA still did really well. You can see the neutralizing titer drop from 6,000 to 2,500. Not a big drop, but a drop. But look at the protein. It went from 2,500 to 250, a 10-fold drop. No deaths in the mRNA group, 30% in the protein group. No 0.5% of the days genital disease in the mRNA group, 20% of the days in the protein group. And again, no shedding of replication competent virus in the mRNA groups, a little bit, 1.4% in the protein. So the mRNA stayed strong, the protein started to fade. Oh, it says challenge at 1 month. It should say, I'm sorry about that. That should say challenge at 8 months. That bottom one is challenge at 8 months. Okay, next slide. So where are we? As we heard in the previous talk, BioNTech is sponsoring this trial. We started talking to them about the same time Dr. Balakantra must have talked to them for pancreatic cancer. Now, our vaccine trial started in humans in December. It's at 3 sites, 2 of them in North Carolina, 1 in Philadelphia, and there's a dose escalation phase. Is the first part of the phase 1 study. Safety and immunogenicity, we expect to have those results probably in about a month to know how well we're doing. So that's very exciting. Next slide. I just wanted to say a few words about what else are we doing. So therapeutic vaccines, a big topic. We're very interested in this. The primary goal, prevent recurrent genital lesions. That's what we're thinking is the most important. What are the challenges? Well, the correlates of protection are not well defined, but the field, and I agree with this, thinks that T cells, probably CD8 T cells, are key. We're using our mouse model to identify potent T cell immunogens, maybe not the same immunogens that are in the prevention vaccine. Maybe we can get away with one vaccine for both, maybe not. We're trying to sort that out. But what we do is our challenge. We infect guinea pigs with HSV2, let them recover, and then we immunize with our candidate vaccine to see if we can reduce the recurrent genital lesions. Now, we're also very interested in HSV1. Our vaccine in preclinical studies, the one that's a prevention vaccine, is excellent against genital HSV1 and genital HSV2. But we're also interested in the non-genital HSV1, herpes encephalitis, herpes keratitis, neonatal herpes caused by HSV1 and HSV2, and fever blisters orolabial. Okay, final slide. Next slide. And just to acknowledge the group, I want to particularly mention Gary Cohn standing beside me. His lab in the dental school, my lab in the medical school have been working closely on this for many years. And at the bottom is NIH and BioNTech for funding and many other people. Okay, thank you very much.
Thank you very much for your presentation. The final member of our panel is Dr. Lorenzo Giaccani from the University of Washington. Dr. Giaccani, your slides are up. I see you on the Zoom.
Excellent. Thank you. I want to thank the organizers for inviting me to discuss work done in my lab in collaboration, of course, with so many other investigators to develop a preventive syphilis vaccine, because, of course, syphilis can be easily treated with antibiotics. Next. So throughout my talk, I wish to discuss why a syphilis vaccine is needed, what, in my opinion, are possible features of an effective vaccine, current and future directions to give us a product that is compatible with manufacturing processes and suitable for clinical trials within the next few years, and finally touch upon new technologies that are being used or developed in order to lead us to this goal of a clinical trial. Next. So there's no doubt in my mind that we need a syphilis vaccine. We just need to look at syphilis epidemiology. And without touching upon the global epidemiology of syphilis, I just want to point out that syphilis has been resurgent in the United States for the last 20 years, as you can see from the graph on the left with data from the CDC. And what's very worrisome is that the rate of infectious syphilis among women in reproductive age is increasing, which leads directly to an increase in the number of cases of congenital syphilis. Next. And you can appreciate that increase from this map. But what's important to say is that congenital infection leads to stillbirth and perinatal death. And an effective vaccine should be able to prevent these very serious outcomes. And at the same time, you can appreciate how such vaccine could be really a game changer for global maternal health, because every year in the world, there are about 1.4 million pregnancies that are affected by congenital syphilis. So how do we get to this vaccine? Next. So the best teacher is, yes, one more thing that an effective syphilis vaccine could do is it could alleviate the consequences of penicillin shortages like the one we are experiencing right now, and finally could limit excessive use of doxycycline now that this antibiotic is available through DoxyPEP to prevent STIs. Thank you. Next. So how do we get to a syphilis vaccine? Well, the best teachers have been the knowledge of syphilis pathogenesis and the biology of its causative agent, Treponema pallidum, subspecies pallidum. So, for example, we know that the majority, unfortunately not all, but most of the pathogen cells that are present in early primary and secondary syphilis lesions are cleared to a process called opsonophagocytosis. Next, please. So this is a process in which antibodies to surface-exposed antigens of the pathogen bind to the pathogen and allow gamma interferon-activated macrophages to come into the picture, ingest and kill T. pallidum cells. And this is very compatible with the fact that Treponema pallidum is an extracellular pathogen. During natural infection, this process is not complete, and a vaccine is should currently try and enhance this process so that we get complete clearance of the pathogen upon exposure to it. Next. So in my opinion, an effective syphilis vaccine should induce opsonic antibodies, should do that in a Th1 immunological environment with plenty of interferon gamma to activate macrophages. And if the immunity that we aim at generating with a vaccine can also target specific pathogenetic processes like dissemination of the pathogen, that would be definitely a major plus. Next, please. If a vaccine is not completely protective, which means it doesn't eliminate completely the pathogen, at least it should very much attenuate early primary and lesion development. And this is because lesions are associated with transmission of the disease. And it should also inhibit dissemination to prevent outcomes such as neurosyphilis and congenital transmission. If those goals are achieved, a syphilis vaccine would be protective for the individual as well as for the community. Next, please. And so the real question is, what antigens then should we target to get to this vaccine? This is a complex question because although a fair percentage of the Treponema pallidum genome codes for the surface-exposed antigens, they are all known to be poorly expressed, not necessarily expressed at any given time during infection, and finally can exhibit a degree of antigenic variability, which is very important to consider if we want a broadly protective vaccine that is not only active against certain strains. Next, please. So again, an effective vaccine should target multiple surface-exposed antigens of this pathogen, and definitely should preferably target conserved epitopes to get to a broadly protective vaccine. Now, before discussing some of the work that we have been doing, I just want to mention that we are not the only group working on a syphilis, on developing a syphilis vaccine. Next, please. I want to mention that there are also, we are based at the University of Washington. Our collaborators are at the University of Victoria, but there are other groups at the University of Connecticut, Duke, University of North Carolina. But today I will focus on the work that we have been doing. So next, please. And I want to start with this publication, which I co-authored a few years ago, where we used a vaccine design based on the 3 antigens that you see in these pictures. We used 2 of them because they were shown to be able to attenuate lesion development upon immunization, and the last one because it was shown to be able to inhibit dissemination of the pathogen. And when we immunized rabbits with these antigens, we indeed attained these results, including a decrease in treponemal burden at the site of a challenge. This vaccine design, however, had limitations, of course. Rabbits were not fully protected. We didn't completely eliminate the pathogen. The adjuvant we used is not approved for human use. And then 2 of the antigens, the one embedded in the membrane in the picture, are very difficult to manufacture. So what is the current strategy to improve this vaccine design? Next, please. So we know that for antigens that are very difficult to express, like the ones in the picture, The ability to protect against infection is linked to surface exposed loops, which are the target of these opsonic antibodies. So the current strategy is to identify which loops of these antigens are really connected to the protection effect that we see, and sort of transplant this loop into a chimeric antigen that can be more easily expressed and delivered. And so this is what the workflow, part of this workflow tells us. And in addition, we are also isolating monoclonal antibodies against these putative loops to evaluate which ones are really opsonic. So to give you a quick example, next please. We, for instance, immunized animals with this large antigen that is called TPRC. And after the immunization cycle, we challenged these animals on their shaved back with the infectious agent. Next, please. And as you can see in rabbits that were immunized, in the picture to the right, you can appreciate how lesion development was attenuated compared to rabbits on the left that were not immunized. And this is one of the correlates of protection that we look at when we challenge immunized animals. Next, please. Additionally, we went on to perform epitope mapping, and these slides want to show that the regions that were particularly immunogenic during this immunization process are highlighted in red on the protein 3D model. So those are the antigens that we are currently looking to transplant into chimeric antigens that can be more easily manufactured. Next, please. And so we have several options. Those include hybrids with the TP751 protein, which is studied at the University of Victoria, and is also another vaccine candidate, the one in particular that was shown to be able to inhibit dissemination of the pathogen. We're also pursuing viral-like particle, and finally, small de novo designed beta barrels that, although share the structure of the bigger antigen, they are more easily produced because of their limited size. Next, please. There are many challenges and opportunities. They all revolve around the fact that we need to identify protective antigens. What are the epitopes within these antigens that are really the target of this protective immunity? We need to understand antigenic variation in vaccine candidates, find adjuvants that are proper for human use, and finally, good delivery systems, which include also RNA vaccines. But last but not least, we need to start thinking about vaccine acceptability and implementation, being this a vaccine for an STI. Next, please. I want to touch upon new technologies. I know I'm running out of time. In a nutshell, those are all technologies that are being used or developed in order to identify these protective antigens and protective epitopes. And we are very lucky to have phenomenal investigators and collaborators at the University of Washington, as well as in the private sectors. Next, please. Finally, I want to acknowledge the many investigators, and this is really the tip of the iceberg, that are every day working to give us a syphilis vaccine. And next, please. And I would like to also acknowledge the funding agencies and their incredible program officers and directors for recognizing the importance of developing a syphilis vaccine and supporting such efforts. Thank you so much.
Thank you very much. And I want to thank all the members of this panel, and we certainly are going to look forward to hearing more from all of you as we get closer to the availability of these in real world rather than in the trial world. have a couple of questions teed up. We'll start with Chris Arisman.
Whoops. Just wanted to thank all the presenters. This isn't a question. I just wanted to specifically thank Dr. Balachandran. My mom died of pancreatic cancer, and so I was very encouraged to read the media reports last spring, and it was an honor for me to hear what you're doing, and I wish you the greatest success. Thank you.
John Iskander. All right, Daniel Hoft.
I'll just echo that great presentations, very important work, exciting progress. As you're working on therapeutic vaccines, of course, you're going into an inflamed environment, maybe globally versus in certain areas. And so that does bring additional safety issues in terms of making that worse, causing problems in the individuals. So my questions are, first, have you seen any of those kinds of problems in your preclinical work or in the human work? What should we be concerned about? And how can we separate the protective effects from the reactogenicity?
Which one of us?
General question for all of you. Anyone can answer, or
I mean, obviously, yes. We are going into hepatitis patients, so we are going in first with a low dose. We will obviously look for hepatic flares. We are not too concerned. These people have already a lot of antigen. Others have used vaccines. But there is a risk. That's why we're doing a phase 1.
Thank you for your question. In the clinical trial that I presented for cancer, one distinction from the traditional interpretation of a therapeutic vaccine, which is given in patients with visible cancer, our clinical trial was given with the primary intent of secondary prevention. So after cancers are removed, you create a vaccine to prevent reoccurrence of the cancer. The concept there being that as vaccines are essentially pro-drugs that require optimal immune health, host health for optimal functioning, the clinical application of such vaccines cannot mirror the history of clinical applications of other cancer therapeutics such as cytotoxics and targeted therapies, which have been given directly to patients with metastatic cancer. One unique challenge with an individualized vaccine, of course, is the fact that each patient's vaccine is its own vaccine. It's an N of 1 vaccine. So, in fact, before our trial, we had extensive discussions with the FDA about how optimally to monitor for safety in such a clinical trial. And in our initial, so based upon these discussions, we decided in abundance of caution to double our sample size to make sure that we will potentially at least allow for the possibility to capture for any patient-specific effects of cross-reactivity of the target antigen to other host proteins. And in our clinical trial, we found a very good safety profile. Only one of the 19 patients had grade 3 toxicity requirement, which was fever, and treated in the hospital quite easily. So we think this has to also do with the general now understanding the good safety profile of RNA vaccines that we now have a lot of information on, in contrast to what we had when we started with.
I can make a very brief comment on the herpes. In preclinical, the models that we're working on, the guinea pig model where you get recurrences, we do not see any enhancement of genital lesions, for example. And we're not in humans yet with ours, but there have been a number of clinical trials that have not succeeded for treatment of genital herpes. And there the problem hasn't been enhancement, just lack of efficacy. So, it's a great question, something for us to keep in mind. I think getting something that works is going to be the challenge, and probably enhancement will be less so.
For the syphilis vaccine, again, I want to highlight that this is a preventive vaccine, not a therapeutic vaccine. In general, at the preclinical level, we don't see any side effects on the animals. But one thing that is that is paramount to improve is the kind of adjuvant, because the REBI adjuvant that we're using right now is not approved for human use because it's too potent, and that definitely causes some discomfort at the site of injection. But I think the moment we switch to a human track adjuvant, those problems will just naturally go away.
I'm not seeing any other questions or comments. I want to thank the members of this panel. You've certainly stirred a lot of interest, and we look forward again to hearing from you down the road. This ends our morning session for the second day of our September NVAC meeting. We will return promptly at 1:15 Eastern Time for our next panel, Respiratory Illness Prevention Priorities for the Season Ahead. Thank you all for participating in our NVAC meeting.
Produced by the U.S. Department of Health and Human Services.